right, so day 2, Mendelian Genetics. Gregor Mendel, this guy right here looking at the plant. He is what's considered to be the father of modern genetics. He pioneered a lot of what we know today about genetics, and this was way back in the 1800s. They didn't know about like microbiology, they didn't know bacteria and all that stuff. They didn't have microscopes. So he, like many other people, were focused on macro things that you could see with your naked eye. You could touch plants, animals, things like that. And Gregor Mendel, he saw these phenomena of, oh, why does this plant have this color flower and this plant has a different color flower? Why do we have things like different hair colors, different eye colors, different heights, skin colors, all this stuff? And he was just like, where does it come from? And everybody else back in the 1800s was like, what are you talking about? You're crazy. Everybody just has these things just because that's the way it is. There's nothing really more to it. And he was convinced, rightly so, that we are products of our parents. Parents give us our characteristics. So what he did is he took some pea plants. Pea plants are perfect for studying genetics because we can observe their traits with our naked eye. They have relatively short lifespans, so we can create lots and lots and lots of new generations relatively quickly. And they're easy to control. So we're talking about experiments and controls. We can isolate certain things with relative ease when we're talking about plants. So Mendel was a prime example of the scientific method. He made a hypothesis. Well, I think that this trait is inherited this way or whatnot. He tested his hypothesis with an experiment. Then he made observations, he collected data, he analyzed that data. And then if his hypothesis was wrong, he would go back, start over, make a new hypothesis, and go through the experimental process again. If his hypothesis was proven correct, he would still do it again and again and again to make sure that it wasn't just a fluke. Doing it over and over with the same results means it will be confirmed. So amazing, perfect example of the scientific method and how that works. So let's take a look at his work a little more specifically. So like I said, he focused on pea plants. He did other things, but his most famous work is with pea plants. He did things like removed stamen anthers to really control the pollination. So it's not like I have my plants out in the wild, the bees and the butterflies and all the other pollinators are just going to do their thing. No, this is an experiment. We want things to be as controlled and perfect as possible. We only want to change one factor. So by doing all of this pollinating and things by hand, so he took like a little paintbrush and took pollen from one and dusted on another one, just to be absolutely sure that the correct plants that he wanted to crossbreed got the correct pollen for creating the new generation. So when he did his experiments, he always started out with true breeding parents. What does that mean? So when we're talking about a parent generation, so the very first generation of an experiment with genetics, things get passed down through generations. We have our parents, they have us, we have kids, they have kids, yada yada. There are many different types of generations that follow that first original pair. That first original pair is called the parent generation. Hey, cool. And we identify that with a P. Makes sense. Parental generation starts with a P. That's how we identify it. In his parental generation, he had what are called true breeding plants. When something is true breeding, that means they are homozygous, either both capital letters or both lowercase letters for their genotypes. Granted, he didn't know about genotypes just yet, but through lots and lots of experimenting with how to pollinate and create all of these subsequent generations and things like that, he was able to pick and create these true breeding or homozygous plants. He raised them, again, many generations. They have a relatively short lifespan and gestation period. And after the parent generation comes what's called the filial. It's Latin You don't really need to remember that. But the way I remember all of these different generation things is the parental generation, P, is the very, very first. Then everything after has an F with a subscript. F1 is the first one generation after the parent. F2, which comes next, is the second generation after the parent. So the Fs stand for filial, first generation of offspring. After he made his F1 generation, he observed some phenomena and decided, hey, what would happen if I took this generation, this new F1 generation, and pollinated it together to create the next F2 generation. So here's another way of organizing this information. Same stuff, repetition, repetition, repetition. So P for parent, we have a parental generation of true breeding pea plants. One is true breeding purple, one is true breeding white. We're gonna breed them together, and what we end up with is that our F1 generation, our first set of offspring. And what he found was 100% of them, every single one of those pea plants was purple. Where did those white flowers go? We started with a white flower. Why aren't we seeing any white flowers in the next generation? This is a strange phenomenon. So he said, okay, I'll take this 100% purple flower first generation of our offspring, and we're going to breed those together. We're going to self-pollinate. So one of these of the F1 generation is going to pollinate with another one from this F1 generation. And what he found was, look at that. We still have purple flowers, but now there's a white flower. It came back. So we say sometimes it skipped a generation. Why would that be? 3/4 of them, 75% of those flowers were purple, but only 1/4 of them were white. That's an interesting phenomenon too. We started with one of each, then we got the first generation, which was all purple, and then the second generation is 3-to-1 ratio. 3 out of the 4 were purple, 1 out of those 4 was white. Interesting. So Mendel then figured out this concept of dominance. Purple ended up being the dominant allele, the dominant phenotype. White is recessive. So because purple is dominant, for our sake, for my sanity, I chose the letter P. P is for purple. Purple is dominant. Capital P gives us a purple phenotype. So we have capital P, capital P. We're going to cross that with a white flower, which is lowercase. We cannot change the letter to W because we can't change the allele letter, but we can change the allele capitalization. So we have our parent generation here. They crossbreed. They get one big P from the purple plant, one little P from the white plant. That gives us a heterozygote for our F1 generation. Since it's heterozygous, we still have a dominant purple allele. That's the phenotype that's expressed. Here's another way to look at it, adding more vocab, repetition, repetition. P for parent generation, true breeding purple flowers. We now know that their genotype is capital P for purple. The phenotype is, again, true breeding, homozygous, purple. We're going to cross that with our true breeding, white, recessive flower plants with their genotype little p. It's recessive, so we have the lowercase p's, and because it's recessive, we have to have both of them as lowercase. We're going to breed them together. We get our 100% purple flowers, and why? Well, look at that. They all are heterozygotes. They got a capital P from the purple flower and a lowercase p from the white flower. Put them together, one of each, heterozygous. They are 100% purple in their phenotype. The dominant purple allele is there, making it show up and canceling out that white. So white is not present. The dominant allele took over to show our purple flowers. Now we're going to self-pollinate our F1 generation, which will give us our 3-to-1 ratio, purple to white. But what do their genotypes look like? This is going to be interesting. Punnett squares are the answer here. Punnett squares. Hopefully this should be slightly familiar, reminiscent of junior high. You should have done something with Punnett squares in junior high, but fear not if you don't remember. We're going to go through it step by step. So you have your, we're trying to figure out our F2 generation genotype here. Our parents are going to be from the F1 generation. We're going to cross-pollinate our F1 generation to get our F2 generation. And traditionally in our Punnett squares, males go on the top, so the sperm, and females or eggs go on the side. For my class, I don't care. Sometimes it doesn't even tell you which one's the male, which one's female. You're going to get the sum results whichever way you put it. In the future, if you pursue a career in biology or take some higher level biology classes, they may care, but for my class, doesn't matter which side the male and female go. All right, for each allele, each letter, looking at the top here, 1 letter per column. Looking at the side, 1 letter per row. Then that was the hardest part. Take what's in the top, take what's on the side, put them together. That's one of your offspring's potential genotypes. Get a big P from the male, big P from the female. We get a double big P. Big P, big P. Next box over, we get a big P from the female, little p from the male. Put them together, that's our genotype. 1 big, 1 little, heterozygous. Moving down to the bottom left, big P from the male, little p from the female, heterozygous again. And lastly, oh look, we get a little p from the male and a little p from the female, giving us our recessive white phenotype. This is how our white seems to reappear or skip that generation. Phenotypes and genotypes can have different ratios. What does this mean? Well, looking at our F2 generation, our genotype, we had 1 of the 4 homozygous dominant, big P, big P. 2 of them were heterozygous. So 50% of them, 2 out of the 4, was heterozygous, 1 big, 1 little. And 25%, or 1 out of 4, was homozygous recessive, little p, little p. So our genotype ratio, if you don't like percentages and you want to reduce it, was 1 to 2 to 1. But our phenotype ratio is going to be different. 75%, or 3 out of the 4 flowers, were purple, and 25% were white. So our genotype and phenotypic ratios are not necessarily going to be the same. Here's the breakdown of the simpler ratio breakdown, 1 to 2 to 1 or 3 to 1. I prefer to use percentages. It's up to you if you give answers to me on your practice and things like that. If you like the simplified ratio or the percentages, it's up to you. Because our ratios can be different, this also means that 2 different organisms can have the same phenotype but have different genotypes. We know this because of dominance. So again, from our F2 generation, we have our homozygous dominant, big P, and our heterozygous dominant, big P, little p. They have different genotypes, but they're both presenting or expressing that purple phenotype. Same phenotype, different genotype. So looking at this flower color on his pea plants, Mendel came upon this first law of heredity, which is called the Law of Segregation. So back way when Mendel was doing his experiments, he didn't know about meiosis yet. They didn't realize that, egg and sperm cells come about differently than other cells. They didn't even necessarily know about cells yet. I don't actually know when they discovered cells. But anyway, he didn't know about meiosis yet, but he still came to this conclusion based on the observations he was making about his pea plants. We know about meiosis. So during meiosis, we know that alleles segregate. This is why we have 1 allele per gamete. We have 1 P from mom, 1 P from dad. Put them together, we get a big P and a big P gives us our genotype, which we'll express as our phenotype of purple, one from each. But the homologous chromosomes, so here we have our 2 homologous chromosomes. They happen to be double-stranded. They're going to separate. We know this. Meiosis 1, we're just separating those homologous chromosomes. Then, during meiosis 2, we're going to split those in half so that now we have an allele, a single allele, for each trait, and it's packaged in its own gamete, whether that's a sperm or an egg. Mendel figured all of this out just with some pea plants. Crazy smart dude. So when does this law of segregation happen? Well, looking at meiosis, we know that those homologous chromosomes separate during anaphase 1, and then we get those sister chromatids at the end of anaphase 2. All right, nitty gritty here, we're gonna get into some Punnett squares. Monohybrid crosses. Looking at our roots, mono means 1. We're going to focus on one characteristic or trait. So like the example we used before, the color of the flowers on his plants. Only looking at one of those traits out of the many different types of traits that a flower plant might have. Height, seed color, all of those different kinds of things, but we're only going to focus on one in a monohybrid cross. So let's practice together. So in our problem here, a heterozygous brown-eyed dog, we'll call this dog 1, breeds with a homozygous brown-eyed dog, we'll call that dog 2. So one is heterozygous, one is homozygous. They both have brown eyes. What are the possible offspring eye colors if brown eyes are dominant? Show both phenotypic and genotypic ratios. So before we can figure out our ratios, we need to make our Punnett square. So what do we know? We know that brown eyes are dominant, it told us that. Makes sense. Our heterozygous dog is brown-eyed. We know the genotype of dog 1. Since it's heterozygous, I chose B for brown. Just makes sense to me. If you want to choose like X or Y or something, that's your choice. But I like to choose the letter that corresponds with the dominant trait. All right, so we have dog 1, heterozygous for brown eyes. Heterozygous means one of each capitalization. Dog 2 says is homozygous for brown eyes. If it's homozygous and it has brown eyes, the dominant trait, both of those letters must be capital. Now we got to put them in our proper spots on our Punnett square. Doesn't matter which one goes where. I just arbitrarily chose dog 1 to go on the top, dog 2 to go on the side. 1 allele per column for dog 1, 1 allele per row for dog 2. That was the hardest part. Now we just match them up. This first one, we get big B from dog 1, big B from dog 2. This offspring has a potential of 2 capital B's. I like to go like the way we read, left to right, top to bottom. If you want to go in a different order, that's also fine. So up next, we're going to get a big B from dog 2 and a little b from dog 1. Note here for best practice. Capitals always come before lower cases. Just the way that it makes things easier to read. In our next Punnett square practice, we're going to focus on more than one trait. And so keeping the capital letter before the lowercase letter makes things a little bit easier to read when you're trying to figure out the phenotypes here. All right, next square, capital B from each parent gives us a double capital B. And lastly, big B from dog 2, little b from dog 1, making sure that capital B comes first. Great, we finished our Punnett square, hooray. Now the last part, we have to figure out our phenotypic and genotypic ratios. So let's start with our phenotypes. Our phenotypes, well, we're looking here, at least one capital B is in every single one of these squares. So that means 100% of our dogs are going to be brown-eyed. Brown is dominant. We're going to have all brown-eyed dogs. Next, we need to look at our genotypic ratios, the letters themselves. Out of our 4 boxes, we have 2 different genotypes. Half of them are capital B, so 50% of them. Again, I prefer personally percentages to the regular simplified number ratios. If you want to do 1-to-1, that's fine too. But again, my personal preference is percentages. And 50% of them are heterozygous, one of each capitalization, big B, little b. So even though our phenotypic ratio is 100%, our genotypic ratio can be different. Questions on monohybrid cross Punnett squares before we move on?
Would you be able to quickly go over the difference between just like a simplified explanation of like phenotype and genotype?
Absolutely. So phenotype is the physical attribute, what is actually being expressed. So in this case, brown eyes is going to be the physical characteristic that we're talking about. In my case, for my hair color, I have brown hair. Based on my parents, my genotype may be something different. So our genotype are the actual letters displayed for our phenotypes here. What is the capitalization of those letters? Is there a dominant one or is there a recessive one? So for our dogs here, a heterozygous brown-eyed dog, so we're looking at dog 1 up at the top here. Heterozygous means it's going to have one of each capitalization. Its genotype is capital B, little b. For dog 2, it's homozygous for brown eyes. Homozygous means they're going to be the same capitalization. Brown eyes are dominant, so we know they're both going to be capital B. Its genotype is big B, big B. If we're using my hair as an example, my mom has brown hair, my dad has blonde hair. I carry that recessive blonde gene. It's just not being expressed because brown hair is dominant over blonde hair. So my phenotype is brown, but my genotype would be heterozygous, big B, little b.
Thank you.
Sure thing. Good question. All right, dihybrid crosses. If you've been zoned out this entire time, this is the one thing I need you to actually listen to. This is super complicated. This is the most difficult part. All right, dihybrid crosses. Other experiments that Mendel did focused on 2 traits and characteristics. Looking at our roots here, di means 2, dihybrid, like our monohybrid. Dihybrid has 2 traits that we're going to look at the same time. So in Mendel's case, when he looked at his pea plant's seeds, he looked at the color of it. So here in my picture, we have green and yellow seeds, and the shape of those seeds. We have some smooth round ones, and we have some weird wrinkly looking ones. So he's comparing 2 different traits. The next slide is going to be really overwhelming, but we're going to take it step by step, break it down. All right, don't forget, starting at the top, we have our parent generation, true breeding yellow round peas. Yellow is dominant, round is dominant. That means for my sanity, I chose Y for yellow and R for round. If you wanted to use other letters, X and Y, whatever, that's your choice. But for me, this makes the most sense. So this is what I put. Because yellow is dominant, I make a capital Y. Round is dominant, capital R. We need 2 alleles for each characteristic, which is why we have 2 Ys and 2 Rs. Looks kind of weird because now we have 4 of those letters instead of just 2, but we're looking at 2 different traits. On the other side, we have a true breeding green and wrinkled pea. We cannot change the letters. We can only change the capitalizations, which is why we don't have G for green and W for wrinkled. We have to stick with the letters we chose for our dominant traits. Lowercase letters are for our recessive traits. In this case, those recessive traits are the color green and the wrinkled texture shape. Because they're recessive, they need to be homozygous. Little y for green, little r for wrinkled. We're going to breed those together, and lo and behold, our F1 generation is all round and yellow. Thinking about our monohybrid pea plants here, that kind of makes sense. Looking at our genotype, we have one big Y from this one little y from the other one big R from one little r from the other. They're heterozygous for each of those two traits. Heterozygous for the color yellow, heterozygous for the shape or texture of round. What would be dominant if your mom has brown eyes and your dad has blue eyes and all your sisters have green and hazel eyes? Yours is brown. Okay, wonderful question. I'm using some traits like eye color as examples because they're things we're familiar with, we can see all the time. But in reality, things like skin color, eye color, even hair color have more than one allele that contribute to determine that color, which is why we have so many different eye colors: green, blue, gray, hazel, brown, different shades of brown, stuff like that. So we even have like heterochromia. So if you notice, like husky dogs, some have like one brown eye and one blue eye, things like that. But just for the sake of simplicity and using examples we're familiar with, I've been using eye color. No worries. Glad you asked it. Excellent question. All right, so our F1 generation, heterozygous. Because it's heterozygous for both traits, all of them are going to be showing the dominant phenotype, yellow and round. Then we take that F1 generation and we're going to self-pollinate. And here are our results for the F2 generation. Out of these 16 little plants that come out of it, 9 of them are yellow and round. Makes sense, they're the dominant traits, should have the most of those. Then we get 3 green and round. Weird, we've never seen that combination. And we get 3 yellow and wrinkled. Another weird combination we've never seen before. And only 1 of our little green wrinkled guy. Weird. When we crossed those heterozygous yellow and round peas, we got 2 new combinations that we'd never seen before. Interesting. So what's happening? If the genes are on different chromosomes, so let's say the gene for color, yellow or green, is on chromosome 3, and the gene for the texture or the shape, round or wrinkled, is on chromosome like 13. If they're on different chromosomes, how do they assort into the gametes? Do they do it together or do they do it independently? If they did it together, it would look like this on the left. We would get one that's dominant for yellow and round. We would get one that's recessive, green and wrinkled, every time. Or if they assort independently, we get this one on the right. We get all the different combinations of capital Y's, lowercase y's, capital R's, and lowercase r's. That's where those 2 new phenotypes came from. All right, this stuff is tricky. Stay with me. You got this. We're almost done. So here we have our F1 generation, our heterozygous for yellow and round. Both of them have the same genotype because they're from the same generation of F1 where everything was heterozygous. If you remember from Math 1, Algebra 1, polynomials foiling, first, outside, inside, last. That's what we have to do here to make sure we get all of the possible combinations of that independent assortment. So let's do it. We're going to start with this one on the left, the blue parent here from the F1 generation. Doing the first one, big Y, big R. Outside, big Y, little r. Inside, little y, big R. And last, little y, little r. We're going to FOIL it, just like polynomials in algebra. With our second one here, happens to have the same genotype, so we're going to have the same foiling here. First, big Y, big R. Outside, big Y, little r. Inside, little y, big R. And last, little y, little r. That was the hardest part. That was the hardest part. Even I mess it up sometimes. And, you know, I've had a lot more practice. Now comes the slightly easier part, where those squares intersect. We just pop in those y's and r's. So we get one y, one big Y from mom, one big Y from dad, one big R from mom, one big R from dad. Here's another note about best practice. Keep the alleles for each trait together. Keep the Y's together, keep the R's together. This will be much more helpful when you're trying to determine those phenotypes. If we did YR, YR, it's a little bit harder to differentiate. And then there's an extra step where you have to count, make sure you have the right Y's and the right R's, but if they're together, much easier to do it at a glance. So we get our totally homozygous dominant for color and shape here, yellow and round. Next one, we get two big Y's and one of each for the R's. Again, first step for dihybrid crosses, keep those alleles together. Keep the Y's together, keep the R's together. Then, like in our monohybrid cross, put the capital letter first if you have a heterozygote, then the lowercase. Next one, we have capital Y, little y, two big R's. And we're going to proceed and go, wait, what is this? Here's our first new mystery phenotype. We got two big Y's, one from mom, one from dad, and two little R's. That will give us a yellow color because yellow is dominant. We have the capital letters here. And the recessive shape or texture, two little R's. So that's how we get that first new phenotype of yellow and wrinkled. We're going to keep going here. We have another yellow wrinkle. It's heterozygous for yellows now instead of homozygous for yellow. And we're going to keep going. And what is that? Here's our second new phenotype that wasn't around before. Looking at our genotype, we have two little y's, so recessive color trait here will be green. And our dominant big R shape for round. That's how we get this second brand new phenotype. We're going to make another one. Look at that, two little y's, and then a heterozygous for round, more round, another wrinkled, another green round, and finally we get our little double recessive green wrinkled seed. So here is our breakdown. So out of these 16 possibilities, 9 of them are yellow and round because those are the most dominant. It will be the most common out of all of them. We get 3 of each, green round and yellow wrinkled, and only 1 green and wrinkled. Pro tip, shortcut. In dihybrid crosses, if you are crossing 2 completely heterozygous organisms, so in this case, both of them are heterozygous for color and both of them are heterozygous for shape. Every single time, you will get the ratio of 9:3 to 3:1 for your phenotype. Every single time. If you have one of them is homozygous and one of them is heterozygous, you're going to have to make the dihybrid cross Punnett square. But shortcut, which is always helpful, if you have 2 completely heterozygous for both traits in both organisms, your phenotypic ratio will always be 9:3:1. All right, almost done here. Last little bit. So after doing these dihybrid crosses where he was looking at the seeds, color, and texture, Mendel came upon this second law, of independent assortment. That sounds familiar. Meiosis, again. Mendel doing some heavy lifting, didn't know about meiosis yet, but he still managed to figure out this law of independent assortment. Different genes separate into gametes independently. What that means is non-homologous chromosomes align independently. So non-homologous, that means in humans we're diploid, we have 2 of each chromosome. So that means if our number 4 chromosomes and our number 18 chromosomes, they're not dependent upon each other. They're going to line up during metaphase 1, whichever way they want. It's not going, ooh, number 4 up there is swapped, so that means I have to swap. No, it's just totally random. If they're different chromosomes that are non-homologous, they're not the same, they're going to align independently from each other. This is only true for genes that are on separate chromosomes. Or, so separate chromosomes meaning like that number 4 chromosome and the number 18 chromosome. Or it can also happen if the genes are on the same chromosome, but very, very far apart. So far apart that crossing over happens frequently. So if we're looking at our double-stranded chromosome, it's like the one with the big X, it's got its tetrads, those arms and the legs. If I have a gene for the color of my seed way up here on this tip over here on this end of my tetrad, and the shape of my seed is way, way down there on the opposite tetrad, they're super duper far apart, the likelihood that they're going to cross over is very high. Independent assortment also works there as well. That's how we get some of that genetic diversity. Where does this happen? Hey, we know that. Independent assortment happens during metaphase 1, wherever they line up, whichever which way. As long as they're non-homologous, they line up totally randomly. The only exception to this law is something called linked genes. We're going to get deeper into this next week. And what this means is when genes are super duper close together on their chromosome. So for example, here, my single-stranded chromosome here, it has 2 genes that are very close together. They don't have that opportunity like we did with the other ones that were super far apart on opposite tetrads to go through crossing over to swap around their genetic information. If they're close together, chances are they're going to be inherited together. This is called linkage. Again, we're going to get super more specific about this next week when we talk about exceptions to Mendel. All right, recap, and then we'll be done for this portion of the class. Law of segregation, we can find this happening during the monohybrid crosses when we're looking at one trait. Each allele segregates into separate gametes. We have 1 allele per gamete, so that when we come to fertilize, we get 1 allele from mom, 1 allele from dad. When we fertilize, create the zygote, our zygote now has 2 alleles. It can be homozygous or heterozygous, dominant or recessive. Law of independent assortment applies to dihybrid crosses or more. So if you want to compare 5 traits, you can do that and you can see the law of independent assortment happening. For my class, we're not going to get any bigger than dihybrid. It gets super complicated and messy. Don't need any more than 2 traits to compare at a time for me. Genes on separate chromosomes assort into gametes independently. So chromosome 5 and chromosome 22 are not dependent upon each other. They're going to just line up along the metaphase plate, whichever way they want. They don't care what's happening with the other chromosomes. And this happens during metaphase 1, which we learned during the meiosis section. The only exception to the law of independent assortment is that linked genes that are very close together on a chromosome. Comments, questions, concerns, clarifications?